In this PowerPoint, we'll continue with the electrical signal creation in neurons and looking at the action potential in more detail. An action potential occurs when the summation of stimuli on the neuron causes the membrane potential to depolarize and reach a threshold of negative 55 millivolts. So remember, the resting membrane potential is negative 70 millivolts. And if there's enough stimuli on that neuron, remember there's a summation of that stimuli, and if there's enough stimuli that depolarizes the cell membrane of the neuron or makes it less negative on the inside of the cell membrane. Remember, we start at negative 70 millivolts. If the stimulus brings the inside voltage of the membrane to negative 55, then we create an action potential. And this graph illustrates an action potential. Okay. All right, so you have to know that number. Threshold is negative 55 millivolts. That's the point reached that will create an action potential. And when these action potentials, APs, are generated in a neuron, most typically, this will be a question, most typically they're created at the axon hillock. Remember, the axon hillock is that part of the neuron where the axon attaches to the soma or the cell body of the neuron, also known as the trigger zone. It's called that because most action potentials, when they're created in the neuron, are created at this point here. Okay, and once that action potential is created, it will then be transmitted down the length of the axon to the terminal ends of the neuron. All right, in the next slide, we'll look at the action potential graph in more detail. You have to know the different parts, different phases of the action potential. But generally, there will be it's a very rapid sequence of events that occurs within the neuron and the cell membrane. And first, you will have a depolarization effect, which then goes into a repolarization effect or phase. And the channels that are involved that we'll look at are voltage-gated sodium and voltage-gated potassium channels that open in sequence. Okay, and we'll look at the detail on the next slide. The other really important point here is the all or none principle. So what this refers to is that if there's enough stimulus on the neuron that it depolarizes the inside of the membrane of the neuron to negative 55 millivolts. Okay, once you hit threshold, then the action potential occurs, and it doesn't matter if there's more stimulus, the action potential will always be the same. As soon as you hit threshold potential, negative 55 millivolts, the action potential automatically will occur, and it's always exactly the same. If there's not enough stimulus to reach negative 55 millivolts, all right, so if the inside of the cell membrane only gets to, let's say, negative 60 millivolts, there's not enough stimulus to depolarize it to negative 55, then nothing will happen. Okay, so it's all or none. Either you get a full action potential or you get nothing at all. All right, let's go through the different phases of the action potential. So you have to know each phase and what's going on in each phase. So we'll start off at rest. Remember, the resting membrane potential is negative 70 millivolts, so that's when a neuron is at rest. And recall that the gates, the types of channels that are active to establish the resting membrane potential are the leaky sodium and leaky potassium channels, and as well the sodium potassium pumps are working to maintain this negative 70 millivolts. So now we're going to stimulate the neuron. Some sort of stimulus is going to start acting on the neuron, and it's going to cause a depolarization. So it's going to cause the inside of the cell membrane to become more positive. So for this to happen, from rest to threshold point here, we have to have either ligand gated or mechanically gated sodium channels opening. Okay, remember, ligand is a neurotransmitter. So if a neurotransmitter binds to that channel receptor, it allows sodium to enter into the cell. So remember, sodium is a positively charged ion, so if sodium channels open up, sodium will start to come into the cell, making it more positive inside the cell. Remember that mechanically gated ion channels are those channels that respond to some sort of a mechanical stimulus, like a painful stimulus or vibration or pressure that we see with receptors. And remember, when those channels open, again, sodium, if it's allowed to enter into the cell, will start to depolarize the membrane potential. It becomes less negative inside the cell, right? If there's enough stimulus occurring to cause this depolarizing potential, and remember there's a summation of those potentials. If there's enough stimulus to cause the inside of the cell membrane to hit negative 55 millivolts, okay, you have to remember that's the threshold potential. You have to know that number, negative 55. At this point, at this voltage, negative 55 millivolts, voltage-gated sodium channels will open in the cell membrane, and sodium will flood into the cell very, very quickly. So this is known as depolarization. So this phase from threshold up to the peak potential, this is called depolarization. And you have to know that's due to the voltage-gated sodium channels opening and sodium flooding into the cell, okay, making it much more positive. We see we get all the way up to about positive 35 millivolts on the inside of the cell. At this voltage, the voltage-gated sodium channels will close, and the voltage-gated potassium channels will then open. All right, so the potassium channels that are responding to this voltage will then open, and now potassium is allowed to flood out of the cell. Remember, there's more potassium inside than outside. So if we open up these gates, these gated channels, potassium moves from inside to outside the cell. So now we're losing all those positive charges from inside the cell, and so the membrane potential very rapidly becomes negative again, goes all the way back down. Okay, and this is known as repolarization, this phase. All right, so repolarization occurs when the voltage-gated potassium channels open and the sodium channels close, and potassium is allowed to flood out of the cell. The repolarization phase will continue, and we actually go into a phase called hyperpolarization, where the membrane potential becomes more negative than resting membrane potential. It can go all the way down to negative 90 millivolts. This is known as hyperpolarization. And at this point, all voltage-gated channels will be closed. So voltage-gated sodium and potassium channels close. And if you can imagine, when that occurs, all that sodium that flooded into the cell is now trapped inside the cell, and all of the potassium that flooded out of the cell very quickly is now trapped outside of the cell. So to be able to get the resting membrane potential established again, okay, to get back up to negative 70 millivolts, we have to get those ion concentrations back to where they should be. So during this phase through here, this hyperpolarization phase, and back up to negative 70 millivolts, it's the sodium potassium pumps that are working to pump the sodium back out and the potassium back into the inside of the neuron so that we can get back to resting membrane potential. Okay, just another graph illustrating the action potential. This will show failed initiations. So remember, we start at resting membrane potential of negative 70 millivolts. We have a stimulus. And what this illustrates is that if there's not enough stimulus, not enough summation of stimuli, and we don't hit the threshold negative 55 millivolts, okay, there will be no action potential. So this is illustrating, remember the principle all or none. Okay, so you'll get asked about all or none principle. That means if there's not enough stimulus to depolarize the inside of the membrane to negative 55 millivolts, okay, don't hit threshold, then nothing happens, okay? If we do hit threshold, then the action potential occurs automatically, and it doesn't matter if there's more stimulus at that point. The action potential will be exactly the same every time, all right? And just remember the different phases. Know the ions involved with each phase. Remember that in depolarization, we've got voltage-gated sodium channels that are opening. Sodium's flooding in. We hit peak potential, and that's when the voltage-gated sodium channels close and the voltage-gated potassium channels now open. and now potassium floods out of the cell. That's the repolarization phase. We go right down into hyperpolarization. At that point, all voltage gated channels are closed, and now remember we need the sodium potassium pumps and lots of ATP to run those pumps to pump the sodium back out and the potassium back into the cell so we can get the proper ion concentrations to establish resting membrane potential so that the neuron can be excited again at that point. There are two refractory periods during the action potential that you need to be aware of. So there's the absolute refractory period and there's the relative refractory period. So refractory periods generally refer to a period of time during the action potential when the neuron cannot generate another action potential in response to a normal stimulus. Okay, so absolute refractory period refers to that point in time from threshold, so when the membrane reaches threshold, -55, goes through depolarization, repolarization back down, and the membrane potential again reaches our resting membrane potential of -70. So through this whole period of time through here, right, this is known as the absolute refractory period, and it's impossible to create another action potential in that same neuron during this period. So this is known as the absolute refractory period. There's no absolute way that you can create another action potential during this time. And a major function of this is to prevent the action potential from traveling backwards, back up the axon towards the soma. Okay, so we want the action potential to continue down the axon toward the terminal ends, and the absolute refractory period allows that to occur. The relative refractory period is that period of time during the action potential from when the membrane potential is back down to resting membrane potential, -70, and hyperpolarization through that area. Okay, so during the hyperpolarization phase, because the membrane potential is more negative through here, you would need much more stimulus, right? Much more depolarization to be able to hit threshold potential if you wanted to create another action potential. So it is possible during the relative refractory period to create another action potential in that neuron. So you would need a larger than normal stimulus or a supra-threshold stimulus, so very large stimulus, because we need to depolarize the membrane much more than normal to get back to threshold. So it's possible to create another action potential during this time, but you need a very large stimulus to do so. So this is known as the relative refractory period. Okay, you're going to get asked about these refractory periods. Make sure you know that the absolute refractory period functions to prevent the electrical signal from traveling backwards. It perpetuates the signal in one direction down the axon. Once an action potential is created, it needs to be propagated or transmitted down the length of the axon. There's two ways that this happens. In unmyelinated axons, remember in the body we have myelinated and unmyelinated axons. So in unmyelinated axons, unmyelinated fibers, that action potential will be conducted in a manner known as continuous conduction. So every length of the way, every step of the way down the length of the axon, you have to recreate that action potential. In saltatory conduction, this is what we see with myelinated fibers. Saltatory means frog-like. So we see that the action potential, once it's created, leaps from node of Ranvier to node of Ranvier. So what we're looking at in this image is in the peripheral nervous system. This is a Schwann cell. So remember, Schwann cells create the myelin in the peripheral nervous system. And if you remember what the cell type is in the central nervous system, it's the oligodendrocyte that makes the myelin. But looking at this image, we've got Schwann cells, and between each of the Schwann cells here, we have a naked area of the axon that's known as the node of Ranvier. So once the action potential is created at the axon hillock, it leaps from node of Ranvier to node of Ranvier in a frog-like manner, and this is very, very quick. So the depolarization of the membrane occurs only at the nodes of Ranvier as opposed to every step of the way all the way down the axon, as you would see in continuous conduction. So This is what makes myelinated axons conduct the electrical signal much faster than unmyelinated. All right, this image again just illustrates the difference between continuous conduction and saltatory conduction. So on the left here, we've got continuous conduction. This is an unmyelinated axon. And once the action potential is created, it has to be recreated, or you have to depolarize the membrane every step of the way, all the way down the axon. Okay, so this is what makes it slower. It also requires more energy to do this, because remember, during the hyperpolarization phase of an action potential, the sodium-potassium pumps are working to reestablish the ion concentration gradients. And so remember, sodium-potassium pumps require ATP to run them. So continuous conduction, you're doing this all the way down the axon. It does take more energy to do that, and it's slower. On the right side, we see saltatory conduction. Remember, saltatory means frog-like. So in this case, once we get the action potential created, the depolarization occurs from node of Ranvier to node of Ranvier. It leaps from node of Ranvier to node of Ranvier. It's very quick, very fast, and this is what makes, again, myelinated fibers so much faster than unmyelinated, and requires less energy, because now you're only using the sodium-potassium pumps are only working at the nodes of Ranvier. Couple of things that will affect the propagation of an action potential. Local anesthetics, for one. So many medications are developed in trying to um, affect or change or block the propagation of an action potential. So for example, local anesthetics such as Novocaine works by blocking the opening of sodium channels. So remember your action potential. If the sodium channels are not allowed to open, let's say you've got a painful stimulus trying to depolarize the receptor membrane, but if the sodium channels are not allowed to open, then there's no depolarization, which means you can't reach threshold value, which means there's no action potential, which means you can't propagate that signal to the central nervous system and to the brain, so that in effect there won't be any perception of pain, or you'll reduce the perception of pain in that area. Cooling as well. So one of the reasons why we instruct patients to cool an area that's been injured, cooling will slow the propagation of the action potentials carrying pain signals, for example, to the central nervous system. So it can also affect the perception of pain, or reduce the perception of pain. Okay, factors that affect the speed of propagation of an action potential. So very simply, the amount of myelin, we already talked about this. Myelinated fibers carry the action potential much quicker than unmyelinated. Axon diameter. So there are different diameter axon with the different neurons in the body. So the larger the diameter of the axon, the faster it carries the impulse. All right, and this is similar to wiring, for example, house wires. So a larger gauge of wire will carry the electrical signal faster. Okay, so it's the same in the human body. Large diameter axons are faster than small diameter axons. And thirdly, temperature. So by increasing temperature, action potentials will travel faster. By cooling temperature, using an ice pack, for example, we're going to slow the propagation of that action potential. Okay, three major types of axons in the body, or fiber types, are A fibers, B fibers, and C fibers. So we'll just kind of highlight major differences between these in terms of how fast they are, how fast they carry the signals. So A fibers are the largest, largest diameter and fastest, and you see 130 meters per second, very quick. They're associated, or they make up, the myelinated somatic sensory or body sensory afferent neurons, as well as the motor efferents to the skeletal muscle. Those are all A fiber, large diameter, myelinated. Also, their A fibers carry fast pain. We'll talk about fast pain in more detail later in the course, but when you think about having a painful stimulus, for example, stubbing your toe, there's always that first pain. It's very quick, it's very sharp. Okay, tells you exactly where it is in the body. That pain is known as fast pain. It's carried in the A fibers. On the other end of the spectrum, we have C fibers. So C fibers are small diameter, unmyelinated, and they're the slowest. So 2 meters per second, that's a big difference from 130 meters per second, carrying that impulse. And we see this with unmyelinated sensory and autonomic motor. Don't worry about the autonomic stuff at this point. We'll talk more about it when we do it later in the course. But C fiber pain, you do have to know this at this point, is involved with or carries slow pain. So again, go back to the example, you stub your toe, that first pain is very sharp, it's very fast, that's carried in A fibers. But you know that about a second later, there's going to be that second pain. That's slow pain, and that's carried in the C fibers. And the slow pain tends to be a more nasty pain, and it's often associated with an emotional response, and it's important in creating memory to that event, etc. So again, we'll do pain in much more detail later in the course. At this point, just know C fibers are the smallest diameter, they're unmyelinated, they're the slowest, and they're also associated with slow pain. A fibers are largest diameter, they're myelinated, they're the fastest, and they're, among other things, associated with fast pain, as well as being motor to skeletal muscle, and also the sensory afferent neurons carrying body sensation. In between these two are the B fibers, so they will be the medium speed. They're myelinated as well. Visceral sensory, also involved with the autonomics. So a lot slower than A fibers, quicker than the C fiber though. So again, it highlights axon diameter. The larger the axon diameter, the faster the impulse is carried. Myelinated is faster than unmyelinated. All right, just an activity to help you learn or to reinforce your learning of the action potential. So get a piece of paper, draw out a graph with the action potential, label the resting membrane potential. You should know that you should know the number, so it needs to be negative 70 millivolts. No threshold potential is negative 55 millivolts. Illustrate the depolarizing phase, the repolarizing phase, the hyperpolarizing phase, the two refractory periods, absolute and relative, and also where in the action potential are the sodium potassium pumps working and why are they working there. And you should know at each phase which ion channels are opening and closing. So remember, we have 4 types of ion channels. We have the leaky channels, we have ligand gated, we have mechanically gated, and we have voltage gated channels. So you should know each of those channel types, where they're functioning at each phase of the action potential.